Everybody, we are learning the human physiology, and today's topic is regarding the nervous system, and we are talking about the synapse. In the previous lecture or the previous videos, we have already seen about the synapse structure, and also we have seen about the synapse properties yesterday only. Now today's lecture is about the synapse part 3, and where we are discussing the functions. But try to understand, this is nothing but a revision of the previous lecture or the videos, because many of the things you have already learned about that. For example, you know what is synapse, you know what is presynapse, what you know postsynapsis, you know the synaptic cleft, you know the neurotransmitter, you know who is removing that acetylcholine ACH. So cholinesterase, this type of the enzyme is there. Then the stimulation is going either in a positive way or in a obstructive way. That means there may be the excitatory or there may be the inhibition. So all these words you know, and this will be repetition of all these words in today's lecture. That is what you have learned is nothing but the synapse transmission or the synapse function. Synapse function or synapse transmission having the same meaning. So I request all of you to write down the previous year question or the probable question on this topic. Describe synaptic transmission for 5 mark SAQ. Describe synaptic transmission SAQ, short answer question, for 7 marks. So minimum answer expected is full 1 page and the previous half page minimum. So 1.5 page is the expected full scope answer expected for this question. So let us start from the slide number 1. I am moving further. So I am moving slide number 2. This is the point in the syllabus 2B. So I request again syllabus point write down 2B. Nervous system is the second topic in your syllabus of physiotherapy. Okay. So there are 3 points in synapse. We have already discussed structure. We have already discussed properties, and we have now on the transmission. Slide number 3. Yes. So give the titles. Excitatory function. Synapse. Give the title in your notebook. Some keywords should be there. Synapse excitatory function. Now don't write anything. Just concentrate on the slide number 3. First we will try what is there. Excitation is a stimulation from presynaptic nerve fibers to either muscle or either the next nerve, because the maximum time we are having the NM junction. NM junction is also probable question for SAQ, not based on this thing, but NM junction is the same thing, neuromuscular junction. That is a synapse, that is a transmission. Okay, so excitatory postsynaptic potential, we have seen this short form. EPSP is the non-propagated electrical potential that develops during the process of synaptic transmission. When the action potential, that is AP, reaches the presynaptic axon terminal, the voltage gated calcium channels at the presynaptic membrane are opened. So what is the main stimulation? Calcium is the main stimulation. Are you getting? For what? Excitation. Excitation is where? On the postsynaptic side. Okay. So which are the keywords? Please write down. EPSP is equal to, write down the same word, EPSP. You should remember this word. EPSP is equal to, write the full form, excitatory postsynaptic potential. Excitatory postsynaptic potential. What is potential? Can you remember? Strength. Correct. Write down. Potential means strength. Okay. On the next line, keywords. How it is developing? AP reaches. Please write down. AP reaches presynaptic axon. AP reaches presynaptic axon. Action potential is like a impulse, is like a current, is like a wave. It is starting from the brain or it is starting from the spinal cord. It is reaching there. Okay. Next, what after AP generation, what is happening in that synapses? Third line in your notebook. Calcium channels opened. Calcium channels opened. Underline the calcium. Underline the word calcium. So calcium channels opened. Now I am moving on to slide number 4. Just wait, don't write anything. Now concentrate on the screen. Slide number 4. Calcium ions enter into the axon terminal from ECF. Calcium ions cause the release of neurotransmitter substance from the vesicles by the means of exocytosis. You remember that vesicles? Vesicles are like bags, and these vesicles contain ACH, and ACH will come out from the vesicles. Is it okay? Then ACH excitatory function pass through the presynaptic membrane and synaptic cleft and reaches the postsynaptic membrane. Now what is there? There is welcoming unit. You remember welcoming unit? Welcome, welcome, welcome. Always receptor. Who are doing welcome in the life? Receptor. Don't forget that. Everywhere there is a receptor. In the nervous system, receptors. In the endocrine system, receptors. Can anybody once again revision? Tell me something receptor problem in the endocrine system. Most common disease. Receptor. Yes, please. Correct. Diabetes mellitus, there is a deficiency of either insulin or insulin receptors. This fact is very clear. So some drugs are stimulating insulin, some drugs are stimulating receptors. When you do the running, when you do the walking, what is happening? Not insulin. Receptor number is increasing. Is it clear? So same word is over here, receptor. ACH receptors are there and they form a joint. Complex is a joint. Which joint? Neurotransmitter plus receptor complex. Neurotransmitter receptor complex causes production of non-propagated EPSP. Common excitatory neurometer in a synapse is acetylcholine. So few important words in slide number 4. Please write down. Due to the calcium ions, comma, release of ACH. Calcium mule, because of the calcium, ACH is released. Okay? Next line. ACH is accepted by receptor. ACH is accepted by receptor. Where is receptor? In the postsynaptic membrane. Write down. Accepted in postsynaptic membrane. Receptors are there, not on this side. Next point. ACH is excitatory neurotransmitter. It will always excite. Stimulation. Excitatory neurotransmitter. In the excitation you write down stimulation, giving the stimulation, making the action more fast, quick. That is excitatory. Stop. That is inhibitory. I am moving on slide number 5. Just wait, don't write anything. Neurotransmitter receptor complex, when it is in the, now we are talking about postsynaptic activity. Yeah? What is happening in the postsynaptic activity? Sodium channels will get open. And these sodium channels, they will start mild depolarization. Changing of the RMP. Where? On that side, postsynaptic side. Okay? So, this mild depolarization is called as a EPSP. This is a local potential, that is a strength in the synapse. Now you write down the activity in the postsynaptic side. Slide number 5, S5. Please write down S5. S5. On postsynaptic side, sodium ions enter into cell body. Sodium ions enter into cell body. Neuron hai na? Into the cell body from the external fluid. They are coming inside into the cell. Which? Sodium ions. Where? In the postsynaptic side. Okay? Next line. Mild depolarization takes place mild depolarization takes place. Mild depolarization takes place at postsynaptic side. At postsynapsis. Membrane. Postsynaptic membrane. Side means membrane. Now I am moving further. Slide number 6. Just wait and see on the slide. Slide number 6. There is the properties of this excitatory postsynaptic potential. Number 1, it is confined only to that particular synapses. Where is that joint? Excitation is not meant. For example, here my deltoid muscle here or the bicep muscle is getting stimulation. So that is related with only my bicep. That current will not go on my tricep. That current will not go on my forearm. So that is localized phenomena. Are you getting? Where we want to stimulate, that part will be only stimulated, not whole body will be stimulated. That is the meaning of that. That is a graded potential similar to the receptor potential and end plate potential. EPSP has two properties, non-propagated, that means there is a end. We want to go up to the only bicep muscle, that is the end. So that is only non-propagated, it will not spread further and only obey all or none law. That means either it will be get excited or it will not get excited. Previously I have explained that. That means it requires, for example, 10 millivolt current. 10 millivolt current. Action potential, impulse. That is a minimum requisite. Then only bicep will start working, contraction. Suppose somebody will say, brain will say that only 7, we have less current, only take 7 millivolt. Just give me few contractions. No, not possible. Either I will do full contraction or no contraction. That is called as a all or none. None means no. Either you will pass or you will fail. No? Sir, I will pass little bit. I will fail little bit. Is it a word? Is a word? No, there is no word like this. I will pass 50%. 50% above is a passing. 49.5 is a fail. 50 is a optimal requirement. No? Same is over here. Either you will get pass in the exam or you will get fail in the exam. Who can answer after your examination if I make a message? How many have students passed? Out of the 30. So some will answer, so 20 are passed, 5 are fail and 5 are incomplete pass. You may have the genius, you may say etiquette or something. No, I am not talking about in that way. Either pass, your final mark sheet shows the either pass or fail. No? So that is the same condition over. So write down slide number S6, properties of EPSP. Properties of EPSP. This is most important point. Properties of EPSP. Write down one after another. Number 1, first point, it is non-propagated. It will not move further. It is non-propagated. And second point, I am sorry, it is a little bit my confusion over here. It is my confusion. I am extremely sorry. We can little bit omit this part about they are not following the all or none law. That means partial contraction is possible, what they are saying. So my all previous skeletal muscles, they are not obeying all or none law. That means you write down partial contraction is possible. Partial contraction is possible in the skeletal muscle. That may be the case in the cardiac muscle. Are you getting my point? Cardiac muscle it is not possible, but in the skeletal muscle it is possible. It is, are you getting? It is totally opposite. That means 10 millivolt current has passed, full contraction is there. In 7 millivolt current has passed, little contraction is possible. It is possible in the skeletal muscle especially. Skeletal muscle, they are following all or none law. Please make it very correct answer. Now I am moving further. Slide number 7. Just wait and see what is there. Significance, that is importance. EPSP is not transmitted into the axon of postsynaptic neuron. However, it causes the development of action potential in the axon. When EPSP is strong enough, it causes the opening of the voltage gated sodium channels in the initial segment of the axon. Now, due to the entry of the sodium ion, the depolarization occurs in the initial segment of the axon and thus the action potential develops. from the action potential spreads to the other segment of axon. I will draw the figure on the blackboard so that it is understood. What they want to tell you, it is a wave, it is an impulse, is a current. The same figure we will continue, yesterday's figure. This is the presynaptic. Now everything has been cleared, ACH is there. Here the receptors are there. Am I right? ACH is coming over here. First event, it is because of the calcium. Is it clear? Here, sodium. It is clear? This much is clear. Now, this is the initial beginning part of that whole neuron. Neuron is going like this. Neuron is going down. The nerve is going down, spreading. But what is happening? They are mentioning the whatever the EPSP they are mentioning, it is a local event. And that AP, this AP will like a impulse, like a wave, it will spread to the other part. But whatever they have described is a local phenomena. In this slide number 7, they want to tell you that whatever sodium ions that we are talking about, RMP has changed, depolarization has changed. That is occurring over here, and then like a wave, then it will cross the next segment, then it will cover the next segment. Are you remembering? I think we have already covered this part. You just go on the remaining previous my slide of nerve in the general physiology. And there is also one topic, propagation. Propagation of nerve impulse. And there you remember I have block type of this, like a bogie of the railway. And then we are talking like this. Are you remembering? The same thing they are mentioning in the different language. They are saying that whatever we have mentioned is about the beginning. Okay? So we will try to write down summarize this slide number 7. So please write down significance of EPSP. Title will be the same. Significance of EPSP. First sentence, dictation. When EPSP is strong, that potential strength is strong, comma, NA channels will open in the initial segment of axon. Next line, on the next line, from here, that means from the initial segment, from here the AP spreads, to other segment of the axon, to other segments, like a railway dabba, they move further like a wave. From here, that initial segment, the AP spreads to the other segment of the axon. Gradually it will move further and further. Current will go further and further. Slide number 8. Excitation has finished. Excitation phenomena has finished. So make a one line blank. Don't make a total line, but one line blank, and then give the title Synapse Inhibitory Function. Inhibitory function. Into bracket you can write down inhibition is like a making the function less. Into bracket please write down what is meant by inhibitory. Making the functions less, or to stop the activity. On the next line you write down the 5 types. First we will write down the 5 types. First type, all as it is you have to write down postsynaptic or direct inhibition. Postsynaptic or direct inhibition. Both the names are important. Then second inhibition is a presynaptic or indirect inhibition. Presynaptic or indirect inhibition. Number 3, negative feedback. Fourth point, feed forward inhibition. Feed forward inhibition. And fifth point is reciprocal inhibition. Reciprocal inhibition. So in the excitation there were no much types, but here there are the 5 types, and we have to explain each type in detail in your examination. Okay. So I am moving further, slide number 9. So first type, you write down one line blank and give the title, postsynaptic or direct inhibition. Postsynaptic or inhibition transmission, and stop writing, then we will see what it is. Okay. This much you know, that this is presynaptic and this is postsynaptic. Okay. Postsynaptic inhibition is the type of synaptic inhibition which occurs due to release of inhibitory neurotransmitter from presynaptic terminal instead of excitatory neurotransmitter substance. This is called as a direct inhibition. Inhibitory neurotransmitter is GABA or the dopamine or the glycine. I think this is very easy to understand, this type at least. Here what previously it was, ACH was there. ACH is excitatory. It will definitely do all the time excitation, contraction. So it is moving, so naturally this part is excitation. Now the neurotransmitter is changed. No ACH. That is an inhibitory type of the neurotransmitter. What is that? GABA. So that is giving direct inhibition. Where? On the postsynaptic type. So only because of the direct change of the original, what you can say, the molecule, the phenomena is occurring opposite. So it is very easy to understand. Neurotransmitter is altogether changed. In spite of ACH, here is a GABA type of the neurotransmitter. Okay. So please write down the explanation. You have given this postsynaptic or direct inhibition. Now write down. When presynaptic membrane is secreting inhibitory neurotransmitters, like there are 3 examples. Previously only one, ACH. Now here we are 3. Number 1, GABA. Just remember this GABA. Don't remember this big name. Afterwards you remember big name. GABA. This is very famous, GABA. Number, comma, dopamine. Dopamine. This is giving you the mental tranquility. Yeah? And glycine. So 3 are important, but 2 more important. GABA and dopamine is always there. So dopamine is always there in the brain, especially when you want to rest, when you want to sleep. Then dopamine is acting because your activity is controlled by stopping the stimulation. Okay. So dopamine and GABA is, at the skeletal muscle level, GABA is important, but in the brain, dopamine is important. Next line, because of these inhibitory neurotransmitters, comma, postsynaptic activity is inhibited. जी काय पुढची ऍक्शन आहे ती बंद पाडली जाते किंवा कमी केली जाते किंवा विरोधी केली जाते. जागृत अवस्थेत असाल, when you are awake, it will make you the calm or cool and sleep. So opposite type of action, then the stimulation is taking place because directly supply is changed. Now further will be little bit complicated. Just wait, slide number 10 and look on the board. Don't write these things. Inhibitory neurotransmitter substance acts on postsynaptic membrane. The phenomena is same by binding with the receptors. So like ACH receptors, GABA receptors are there. Dopamine receptors are there. So every time receptor binding is there, then transmitter receptor complex open the potassium channels. Only difference, previously it was sodium channel, here we have the potassium channel opening. So this much line you can write down. Action of GABA on the next line. Action of GABA, underline dash. Action of GABA, underline dash. In postsynaptic membrane, in Post-synaptic membrane, potassium channels open. Make the underline to potassium. Potassium is a K plus. Potassium channels open. That is the most important thing you have to remember. Next, we will go on the slide number 11. Just wait and see what is happening. Now the potassium ions, which are available in plenty of the cell body of the postsynaptic neuron, move to the ECF. Potassium will move out, ECF. Simultaneously, chloride channels also open and chloride channels move inside the cell body of postsynaptic neuron. The exit of potassium ions and influx of the chloride ions cause the more negativity inside, leading to the hyperpolarization. Now the students are understanding why I am saying that nervous system understanding is little bit complicated. Now slowly there is a complicated activity has started in your brain. Oh my God, this is that. But that's why this dictation will be helpful. If you go at home and read quite calmly 2, 3, 4 times, you will automatically understand. Some activities are more there because nervous system we are talking about, brain we are talking about. So naturally, previously it was sodium channels. Now here the potassium is going out, chloride is coming in. Chloride is always negative, Cl minus. You know that. Huh? Sodium is positive, potassium is positive. That is okay. But more negativity is coming inside. Are you getting here? More negativity. That is, in this case, previously it was only AP, depolarization, change of RMP to depolarization. But here the phenomena is different. Here hyperpolarization is taking place. Are you getting little? Okay. Don't take tension. Now write down. Next line. The same phenomena in postsynaptic membrane. Comma, I am making it a simple. In postsynaptic membrane, negative chloride ions are entering into the cell. Negative chloride ions are entering into the cell. Full stop. So, hyperpolarization is taking place. Hyperpolarization taking place. Make an underline to hyperpolarization. Hyperpolarization. You remember the basic 3 figures that we have drawn many lecture back. 3 types of the squares were there. Right? First was RMP, then depolarization and then hyperpolarization. Outside plus, inside negative, RMP. Outside negative, inside positive, depolarization. Am I right? Outside 5 positive but inside 10 negative. If you remember basic these things, all things are very easy. So you remember the basic thing. Don't try to read each and every line of the PPT. Basically concentrate on your notes, and by these keywords you prepare your own notes by proper understanding. Because sometimes this is a semolingam parts, so it is a complicated words. I am making this little bit assimilable type of the dictations. Okay. Now we are moving further. Slide number 12. I think this is the second type. Am I right? This is the second type of inhibition. So write down second type. Please write down second type. Give the full name so that it will not misunderstanding. Presynaptic or indirect inhibition. Presynaptic or indirect inhibition. First we will try to see what is there in the slide 12 and then we will try to make a summary of that slide. Okay. Every time we are making 1 or 2 sentence summary. Now see, it was previously simple thing. In spite of the excitatory ACH, we have the dopamine or we have the GABA. And so whole things are changing. But here, what they are writing, what is happening it is happening in the presynaptic level. Presynaptic inhibition occurs due to the failure of presynaptic axon terminal to release sufficient quantity of ACH. We are excitatory ACH, na? So sufficient quantity of ACH, it is called as indirect inhibition. Presynaptic inhibition is mediated by axonal synapse, only that places. Axonal synapse. We have seen the different types of synapses. No? It is prominent in spinal cord and regulates the propagation of information to higher centers in the brain. So don't remember all these things. This is little bit complicated. I will make it a very simple. So please start dictation on slide number 12. What is presynaptic or indirect inhibition? Please start writing. This inhibition occurs when presynaptic axon is not releasing sufficient quantity of excitatory neurotransmitter, that is ACH, full stop. This is the only essence. You must remember this basic thing. In spite of 100% ACH, there is only 50% ACH. And in that way, that is inhibitory. Actually, it is not inhibitory, but partial inhibition is there. For example, I am expected to take the 10 lectures in a month approximately. And if I cannot take the 10 lectures, it is partially inhibition in your study. Whatever the topics we should cover in the 10 lectures, it is not covered because I was absent, I was ill. So it is exactly not the opposite, but it is something like an obstruction to the excitation. So that is a partial, so that it is word is a indirect inhibition. It is not direct inhibition. Are you getting something? So it is simple sentence, you must write only this thing, that there is an insufficient quantity of ACH. Okay. Now I am moving on slide number 13. Third type, please write down, third type of inhibition. Please give the title, full title, Renshaw cell or negative feedback inhibition. Negative feedback inhibition. Negative feedback inhibition, slide number 13. Just wait and we will try to understand what is there. Negative feedback inhibition is the type of synaptic inhibition, that is very clear. But the main cause, what is the main cause of this inhibition is Renshaw cell. These are the specific cells. We will see the details of that. But this inhibition occurring not because of transmitter. This inhibition is occurring because of the specific cells. And what are the name of the cell? Renshaw cells. Is it clear? Now next. Renshaw cells are small motor neurons present in anterior grey horn of spinal cord. You remember the grey and white matter? Yes. Grey is a collection of cells, neurons, and white matter is a collection of fibers. Am I right? So grey and white. Okay. These nerve fibers arise from the motor neurons in the anterior grey horn of the spinal cord and reach the effector organ of the muscles. There is continuation in the further slide. So you write down the summary of this slide, slide number 13. Please write down dictation. This inhibition occur due to what we have seen, due to Renshaw cells. That's it. Due to Renshaw cells. Into bracket, write down what is that Renshaw cells? Into bracket, small motor neurons. Cells means small motor neurons. Bracket complete. Is it okay? That much understood? Now we will go further. Slide number 14. Just wait and we will see what is that Renshaw cells are doing. Renshaw cells are stimulated. In turn, these Renshaw cells sends inhibitory impulses to alpha motor neurons so that the discharge from motor neuron is reduced. In this way, Renshaw cell inhibition represents a negative feedback mechanism. Now, this point, specifically spinal cord and this discharge etc. alpha neuron, this we will come across when we have the second last or last topic of nervous system, which is called as a stretch reflex. So at this moment you take only deep breath and forget about that alpha motor neuron, how it is inhibited. Only you have to write down that Renshaw says inhibitory impulse to alpha motor neurons. Only this much sentence you write down. Renshaw says sends. Here neurotransmitter is not there. Renshaw cells are there. They sends inhibitory impulse. Same impulse, current, impulse to alpha motor neuron. That's it. To alpha motor neuron. Forget about all other sentences. Now I am moving further. Slide number 15. Next type of the inhibitory function. Fourth, please write down title at least. Feed forward inhibition. Feed forward inhibition. And just wait. We will try to understand first. Slide number 15. Now, feed forward synaptic inhibition occurs. First, it is occurring in the cerebellum. It is not again, neuromuscular is not connected. It is related with the cerebellum. It controls the neuronal activity of cerebellum. So, this inhibition is related only with the cerebellum and activity of the cerebellum. Do you remember the main function of cerebellum? Main function of cerebellum, yesterday only I have mentioned. To balance in the sitting, balance in the standing, balance in the walking, running, etc. Okay? Now, during the process of neuronal activity in the cerebellum, stellate cells and basket cells, which are activated by granule cells, forget about that, inhibit the Purkinje cells by releasing GABA. This type of inhibition is called as a feed forward inhibition. So forget all the sentences. I will give you only two sentences, that much you write, that suffice the matter, okay, for examination. First sentence. This inhibition is occurring in cerebellum. Occurring in the cerebellum. That's it. Next sentence. This inhibition is occurring due to GABA neurotransmitter. This is the name of the neurotransmitter. This is occurring due to the activity of GABA. And where it is occurring? In the cerebellum. Bas. That is sufficient. This inhibition activity is related with the cerebellum, occurring due to the GABA. What is that? Feed forward inhibition. Don't get into the detail, why this is the name, why that is. No, we have to mug it up and write it down in the exam, just passing. Okay? First round should be only passing. Now next, slide number 16. Please write down the title. Fifth part, reciprocal inhibition. This is also simple to understand. Reciprocal inhibition. Wait and look at the board, slide number 16. Very simple to understand. Inhibition of antagonistic muscles when a group of muscles are activated is called as a reciprocal inhibition. It is because of the reciprocal innervation. I will just check whether it is a continued further or not. No, this is something practical significant. But this is very easy. We are talking about bicep and tricep. When bicep is contracted, tricep is little bit relaxed. Antagonistic muscles are always there in the body. Try to understand. So this type of reciprocal inhibition is occurring with relation to the antagonistic muscles. So write down one sentence only. Inhibition of antagonistic muscles, comma, when a group of muscles are activated, full stop. When a group of muscles are activated. If any student is not knowing what is antagonistic, they can write down, antagonistic means opposite. Don't forget, antagonistic means opposite. Good and bad. These are the antagonistic words, opposite words. Now the next part is optional. Those who wants to get the nice to know or desirable to know. Now this is the practical importance of these things. We will try to write down the summarize one sentence only. You give the title at least. Significance or importance. Significance or importance. Significance or importance of synaptic inhibition. Just wait. Now we will try to see what is there in the slide number 17. Synaptic inhibition in the central nervous system. Can anybody tell me what is central nervous system? Which parts important are there? Yes. Correct. Brain and spinal cord is CNS. Can anybody by chance tell me which two things are involved, which two systems are involved in PNS? Peripheral nervous system. Two systems are there. PNS. Come on, be fast, be quick. Peripheral nervous system is okay. What is there? Two things are there. Raise your hand. Yes. Somatic nervous system, which is related with, just wait, which is related with, somatic nervous system related with, skeletal muscle. Don't forget, somatic nervous system is related with skeletal muscles which are on the hands and legs. And second, peripheral is a autonomic nervous system. Sit down. Can anybody tell me, different people should come forward, two types of ANS? Please come on. Two types of ANS. Different people. Yes.
Sympathetic and parasympathetic.
She is correct. Sympathetic nervous system and parasympathetic nervous system. Sit down. Tell me only one function of sympathetic nervous system. Just raise your hand. Sympathetic nervous system. Yes. Energy expenditure by which response? What is the response? What? Yes. Yes. Fight or flight. FIGHT and flight. Fight or flight. Fighting means like this, and flight means to run away from the situation. That is sympathetic. And parasympathetic is doing, please raise your hand, what is parasympathetic is responsible? Yes? Energy conservation by how? How? Yes? Yes, by sleeping. In the sleeping, parasympathetic is more acting. Remember, in sleeping condition, meditation, parasympathetic is more acting. So, this was our yesterday's talk, and now, here is the word synaptic inhibition in CNS limit the number of impulses. Impulse means current. Impulse means wave. Impulse means action potential. All these words should be remembered. Impulses going to the muscle and enable the muscle to act properly and appropriately. Because if it is a bombarding, so the tremors will start. In spite of the contraction, muscle will start twitching or the tremors. So twitching and tremors is abnormal. To control that contraction process, there should be also some inhibition. This is the first important part. Thus, the inhibition helps to select exact number of impulses and to omit or block the excessive one. Very simple to understand. When a poison like strychnine is introduced into the body, it destroys the inhibitory function of synaptic level, resulting in continuous and compulsive contraction even with slight stimulation. Strychnine is a poisoning by a typical herb, poisonous herb. Strychnine is a poison. What is that doing? That is doing destroys the inhibitory function, and so twitching will start. When patient eats strychnine, poisoning will be there, the cramps will be there, muscle cramps. Why? There is no inhibition. Then in nervous disorder like Parkinsonism, the inhibitory system is impaired, resulting in the rigidity. So there are two cases they have mentioned. So you please write down significance of synaptic inhibition. It helps, this inhibition process, it helps for the proper action of muscle, for the proper action of muscles. Proper action of muscles. Make the underline to muscles. On the next line, in strychnine poisoning, please write down this word carefully. In strychnine, slide number 17, strychnine poisoning, poisoning, is a toxin. Strychnine poisoning, comma, inhibitory function stops. Inhibitory function stops. Inhibitory function stops. Full stop. Then there is a continuous and compulsive contraction, at muscles. Next line, in Parkinsonism, this is the name of the disease of the brain. In Parkinson or Parkinsonism, comma, inhibitory system is damaged. Inhibitory system is damaged. Full stop. Inhibitory system damaged and muscles become rigid. Muscles become rigid. Full stop. So my dear friend, this is the end of the synaptic function or synaptic transmission topic, and we will take a break and we will continue in the next lecture. Thank you. Just wait. Which is related with? Somatic nervous system related with?
Body.
Skeletal muscle. Don't forget. Somatic nervous system is related with skeletal muscles which are on the hands and legs. And second, peripheral is a autonomic nervous system. Sit down. Can anybody tell me, different people should come forward, two types of ANS? Please come on. Two types of ANS. Different people. Yes.
Sympathetic and parasympathetic.
She is correct. Sympathetic nervous system and parasympathetic nervous system. Sit down. Tell me only one function of sympathetic nervous system. Just raise your hand. Sympathetic nervous system. Yes. Energy expenditure by which response? What is the response? What? Yes. Yes. Fight or flight. FIGHT and flight. Fight or flight. Fighting means like this, and flight means to run away from the situation. That is sympathetic. And parasympathetic is doing, please raise your hand, what is parasympathetic is responsible? Yes? Energy conservation by how? How? Yes? Yes, by sleeping. In the sleeping, parasympathetic is more acting. Remember, in sleeping condition, meditation, parasympathetic is more acting. So, this was our yesterday's talk, and now, here is the word synaptic inhibition in CNS limit the number of impulses. Impulse means current. Impulse means wave. Impulse means action potential. All these words should be remembered. Impulses going to the muscle and enable the muscle to act properly and appropriately. Because if it is a bombarding, so the tremors will start. In spite of the contraction, muscle will start twitching or the tremors. So twitching and tremors is abnormal. To control that contraction process, there should be also some inhibition. This is the first important part. Thus, the inhibition helps to select exact number of impulses and to omit or block the excessive one. Very simple to understand. When a poison like strychnine is introduced into the body, it destroys the inhibitory function of synaptic level, resulting in continuous and compulsive contraction even with slight stimulation. Strychnine is a poisoning by a typical herb. poisonous herb. Strychnine is a poison. What is that doing? That is doing destroys the inhibitory function, and so twitching will start. When patient eats strychnine, poisoning will be there, the cramps will be there, muscle cramps. Why? There is no inhibition. Then in nervous disorder like Parkinsonism, the inhibitory system is impaired, resulting in the rigidity. So there are two cases they have mentioned. So you please write down significance of synaptic inhibition. It helps this inhibition process. It helps for the proper action of muscle, for the proper action of muscles. Proper action of muscles. Make the underline to muscles. On the next line, in strychnine poisoning, please write down this word carefully. In strychnine, slide number 17, strychnine poisoning, poisoning, is a toxin. Strychnine poisoning, comma, inhibitory function stops. Inhibitory function stops. Inhibitory function stops. Full stop. Then there is a continuous and compulsive contraction, at muscles. Next line, in Parkinsonism, this is the name of the disease of the brain. In Parkinson or Parkinsonism, comma, inhibitory system is damaged. Inhibitory system is damaged. Full stop. Inhibitory system damaged and muscles become rigid. Muscles become rigid. Full stop. So my dear friends, this is the end of the synaptic function or synaptic transmission topic, and we will take a break and we will continue in the next lecture. Thank you. Continuous and compulsive contraction at muscles. Next line, in Parkinsonism, this is the name of the disease of the brain. In Parkinson or Parkinsonism, comma, inhibitory system is damaged. Inhibitory system is damaged. Full stop. Inhibitory system damaged and muscles become rigid. Muscles become rigid. Full stop. So my dear friends, this is the end of the synaptic function or synaptic transmission topic, and we will take a break and we will continue in the next lecture. Thank you. The end of the synaptic function or synaptic transmission topic, and we will take a break and we will continue in the next lecture. Thank you. Thank you.